Chapter 14, Segment 1, pages 433 to 439. Biological rhythms range in length from seconds to years, although daily rhythms are the most studied, which is why this chapter focuses on daily rhythms. So, many animals show daily rhythms in their activity. Circadian rhythms is the name we give these daily rhythms, and they actually repeat about every 24 hours. Many animals are either diurnal, so they're active during the day, or nocturnal, so they're active during the night. And almost every physiological measure changes cyclically over these 24-hour circadian rhythms. And using a biological clock, animals exhibit precise circadian rhythms that vary in time by just mere minutes. So it is very accurate, which is impressive because this is not a mechanical clock. This is a bunch of chemical reactions going on within every cell, which has to communicate with its neighbors and occurring over your entire body. It's pretty accurate, so it's very impressive. The periods, or the times between two ends of these successive cycles of circadian rhythms, changes over time. Oops, I skipped a bunch. Circadian rhythms are generated by an endogenous clock. Despite external lighting conditions, a hamster in captivity has a similar daily rhythm to one that experiences real night and day, suggesting the presence of an internal clock. Circadian rhythms also change slightly over time with use of internal lighting until the hamster is eventually active during the actual daytime. This lack of adherence to actual daytime or nighttime cycles, though, is known as free running. The periods or times between two ends of successive cycles of circadian rhythms changes over time until it does not align with the Earth's rotation and thus becomes characterized as free running. Internal clocks are essentially calibrated to light and thus shifting the period of light eventually affects the activity of an organism, such as a mouse. And this is a process that is called a phase shift. The light in this case that the mice use to calibrate their activity to is an example of a Zeitgeber, Zeitensteinen, Uffensteinen, German noises. An example of a phase shift that occurs in humans due to light is the jet lag experienced when a human lands in a separate time zone after a flight. Circadian rhythms allow animals to anticipate changes in the environment. So circadian rhythms, essentially, may be viewed as an organism's method to adapting to its environment, basically by anticipating future conditions based off of patterns and external cues. So, it's like your body is being very smart. The hypothalamus houses a circadian clock. The endogenous clock that controls circadian rhythms can be found in the suprachiasmatic nucleus, or the SCN, of the hypothalamus. Transplants prove that the SCN produces a circadian rhythm. So some hamsters exhibit free-running periods shorter than 24 hours, rather than the approximately longer than 24-hour periods of most others. So most mice are expected, when free-running, to have periods that last longer than the usual 24 hours. But these mice in particular, for some reason, have ones that are shorter. And researchers identified the cause of this discrepancy as a mutation that they called tau, since the offspring of these mice also had it. So this is a gene that they've been able to pinpoint. And so transplanting the SCN of an embryonic mouse of one of these tau gene having mice into the hypothalamus of a mouse that doesn't have it and has a regular more than 24-hour free running period will cause this mouse actually to exhibit the free running period that is shorter from the donor mouse. So this can actually change the behavior of the mouse depending on the suprachiasmatic nucleus that you put into it. And this actually works in reverse. You can take the SCN of a normal mouse and then expect to see the above the normal 24-hour free running period within this mouse, which actually, because of its mutation, has one that's less than 24 hours. Even when blocking the implanted SCN from sharing synapses with the surrounding hypothalamus by encapsulating it in polymer, it still works, indicating that actually it has a lot to do with chemical signals as well. In mammals, light formation from the eyes reaches the suprachiasmatic nucleus directly. So the pathway that entrains circadian rhythms to light-dark cycles varies depending on species. Let me fix that real quick. So in some amphibians, the pineal gland actually is directly sensitive to light. So it kind of acts like a third eye. Light will go kind of hit the head, and there's a very thin space, but a thin piece of the skull basically between the pineal gland and the light, so it actually reaches it directly. So in mammals, though, light formation reaches the SCN through the eye. Certain retinal ganglion cells send their axons along the retinohypothalamic pathway, which splits off at the optic chiasm to synapse directly with the suprachiasmatic nucleus. So instead of using rods and cones, these retinal ganglion actually use melanopsin, which is its own photopigment, and that's what makes them sensitive to light. So mice that lack rods and cones, therefore basically being functionally blind, actually still entrain their behavior based on the light around them, which shows you that melanopsin is quite independent, and these mice, despite being blind, can still change their circadian rhythms depending on the light around them. So the photopigment is actually melanopsin. It's actually most sensitive to light frequencies within the blue range, which actually makes sense because when you think of daytime, you have these bright blue skies. So daytime light would be associated with the vast blue sky that's overhead. So that makes a lot of sense. Circadian rhythms have been genetically dissected in flies and mice. So Drosophila flies with a mutation that disabled the period gene no longer exhibited rhythmic activity in many experiments. But Drosophila have several genes that affect circadian rhythms, and mammals have several versions of each of these genes. So obviously, Drosophila are a much more simplified version when you're looking at the nervous system of mammals. So although they are very similar, mammals have a much more complicated system, but it is very similar in the grand scheme. So these genes actually provide a molecular clock. So this clock causes protein production between cells to occur cyclically. So I'm talking between each individual cell. Cells in the suprachiasmatic nucleus make two types of proteins, clock cells and cycle cells. And they bind together to form a dimer, which is just a name for two molecules that are bound or bonded together. But then this dimer binds to the cell's DNA to promote the transcription of two other genes, period or per, and cryptochrome or cry. And these two proteins also form a dimer, which in turn inhibits the expression of the clock and cycle genes. So this kind of acts as a negative feedback. The more clock and cycle genes you have forming into dimers, the more you get of these dimers between the per and cry genes, which actually in turn, causes it to happen less. But thanks to the degradation of chemical modification, the degradation or chemical modification of clock and cycle genes, the inhibition decreases and the cycle repeats. And this entire process occurs over the course of these 24-hour periods, which would be the cause of these 24-hour circadian rhythms of the suprachiasmatic nucleus. And communicating through electrical synapses, each neuron synchronizes with one another to create consistency in the molecular clock across the body. And almost every cell in the body actually has the same molecular clock, allowing the suprachiasmatic nucleus to maintain synchronized circadian rhythms all over the body. And in Drosophila, light passes into the brain cells to degrade cry proteins in order to synchronize the molecular clock. But of course, in mammals, it's different. Light info gathered by melanopsin is in the retinal ganglion cells, travels through the retinohypothalamic tract, causing the release of glutamate in the suprachiasmatic nucleus. And glutamate actually indirectly promotes the production of PER proteins, thus shifting the period of PER production, which is what causes these phase shifts. So after your jet lag, this is what occurs in you. And when you move, a hamster from normal circadian rhythms to the circadian rhythms in your lab, this is what occurs.